Hey guys, we are looking at chapter 12. Today we're going to start this chapter and continue our unit on genetics here with this chapter. The way this one's going to work this time, though, for your next test, we're going to combine chapter 12 also with chapter 15, and we're going to actually kind of do some things a little bit in a disorganized way, if that makes sense. So we're actually going to start today with section 12.4. That's not a mistype on the agenda. We are going to do that one first because it actually follows a little bit more closely with what we were doing in chapter 11 prior to this with mitosis and the cell cycle. So we're going to start here, work our way through 12.4, step back and go into 12.1, 2, 3. And then we're going to tie in a little bit with the first couple sections of chapter 15. All right. So it'll be a little bit of time before we get to our next test. But this is really kind of where we get into the heart of all things genetic. All right. So chapter 12, again, intro to the actual study of genetics. And again, we're going to start with section 12.4, which is called meiosis. So you might have learned about this in the past and perhaps even learned to call it meiosis, which is totally fine. There is no one right way to say it. I learned it meiosis, and so that's the way I'm going to continue to say it, but you're welcome to call it meiosis if that's what's most comfortable to you. Okay, so section 12.4, meiosis, go ahead and jot that down. And in this first, section 12.4, but actually the first section that we're going to talk about here in chapter 12, we've got 4 objectives. So we're going to look at genes and how they relate to chromosomes and how many we find in adult organisms. We're going to look at the stages of meiosis, which are going to very closely resemble what we did in chapter 11 with mitosis. And we're going to look at differences and similarities between those two processes, and then also how we can inherit 2 alleles together that are coming from different genes. All right. So in starting us off here in chapter 12, we need to kind of look at the broader picture for a minute. So what we spent a lot of time with in chapter 11 was in talking about body cells. Remember, we said there are 46 total chromosomes in each of your body cells. And I mentioned at the time that your body cells, that term, refers to everything in the body except for sperm or egg. Okay, so except for our reproductive cells. So in this case, we're looking at here, in addition to that, body cells are what we call somatic cells. So this is going to be a new vocab term for most of us. And soma means body. That's where we get that root. And so anything in your body that contains those 46 chromosomes is what we would refer to as a somatic cells, as a somatic cell. So somatic cells come from mitosis and the normal cell cycle. In comparison to that, when we think about reproductive cells, those cells that we use to actually produce offspring, those are what we call gametes. And those gametes come from the division of cells that we call germ cells. Now, germ cells are what we find in ovaries, and we find them in the testes. Those are our reproductive organs, if you're a female or a male. And in those reproductive organs is where we're going to actually create gametes. And we do that through the process of not mitosis, but meiosis. So in other words, we make somatic cells through mitosis. Those are your body cells. We make gametes, or what we call sex cells, or sperm and egg, if we really want to be specific, through meiosis occurring in our germ cells. All right. So when we think about kind of this setup here, right, we can already see some of the similarities that we're going to look at between mitosis and meiosis, in that both are going to create cells and both are dealing with chromosome numbers. It's just the cells that they're going to make here are going to be used for 2 different purposes, right? Our goal in sexual reproduction is to create unique offspring, and a lot of that comes from the gametes, or the sex cells, the egg and sperm, that we create through meiosis as occurring in the germ cells that make up these reproductive organs. And so the sex cells, the gametes that we create here, are not going to be all the same. Unlike what we saw in mitosis, we're going to see differences occurring here in the sperm and egg cells that we create through this process. All right, so meiosis doesn't create identical cells, but mitosis does. All right. So if we look a little bit closer at what makes up these different cells that we find in all humans and most other animals and plants and other eukaryotes, we know that in humans specifically, we have 23 pairs of chromosomes. The image that you're seeing here on the right is called a karyotype. We'll talk about those a little bit more in terms of what they represent as part of this chapter and also chapter 15. But this karyotype that we're looking at here, you can see each of these little lines that appears to be kind of lit up and glowing in this darkness is actually one single chromosome. These are chromosomes that have not gone through synthesis, so they've not been copied, but they are condensed. So what this means is that we've got a scenario where probably this image was taken during mitosis and shortly after the sisters had been kind of pulled apart, all right? And so the chromosomes we see here are still condensed. We can obviously see them in this image, and the way that they're organized here, they're all numbered 1 through 22, except for this final pairing here, which is X and Y. And X and Y belong to a different kind of category. But the ones that are numbered here are always organized by height. We all have a pair of chromosome 1, we all have a pair of chromosome 2 and 3, and so on. And again, in terms of what we inherited from our parents gave us one of each of these types. So mom might have given every chromosome here on the left, and dad might have given every chromosome that's appearing here on the right for each of these pairs. Now, we don't know that for sure, right? And it would be hard to figure that out. But, we can assume that that might be true to just kind of understand what this diagram is showing us, all right? So we have 22 pairs, and these pairs are called homologous chromosomes, all right? Now, I'm saying 22 because these are the ones that we number, all right? They came from two different places. They have the same genes. They have the same structure. But again, their origins are different. One came from mom's side of the family over here. One came from dad's side of the family over here. And dad and mom came from two separate families. So we're looking at the combination of genes from two different kind of ancestral lines, okay? So their origins are different. They do contain the same genes, meaning we may have a gene for eye color here on this chromosome number 2, which would also be present here on this other chromosome number 2. But the difference could be maybe dad donated the gene or the allele for brown eye color, and maybe mom donates the gene or the allele for blue eye color. And we'll talk about what alleles are if you've not heard of that term before. Okay, but we call these homologous because they're essentially the same general shape, structure, genes. What differs is where they originally came from, whether they came from mom or from dad, okay? Of these chromosomes 1 through 22, we call them autosomes because they contain genes for normal body traits. Those are the things that you think about when you look at a person on the surface, right? Hair color, eye color, height, weight. Some behavioral things can be coded for in that. Freckles, the ability to roll your tongue, whether or not you have dimples, a widow's peak in your hairline. Like there's so many different types of cool human traits that we could talk about with this. But the majority of those are what we call autosomal because they're found on any of the chromosomes here that are numbered 1 through 22. The one exception to that, this final pairing that I said is not numbered, but here's represented as an X and a Y, is what we call our sex chromosomes. And for our sex chromosomes, we know every human has to have at least one X chromosome to be able to survive because X does lots of things. But really, the combination of these two is what determines gender, right? Like biological basis of gender. So if you have an X and a Y, that would be male specific. If you have an X and an X, that creates female offspring. Okay. Now there are some differences that can happen with that, and we'll talk about that in chapter 15. What happens if those numbers are off? But for our purposes right now, make sure you jot down with this that XX is female and XY is male, and I'll jot that over here too. So XX female and XY is going to represent a male, what we call genotype. Okay, so male genotype means we're looking at the genes that create a specific trait. Okay, so make sure, again, we jot that down as part of this. Okay. All right, so moving on from that. When we talk about our body cells, the ones that we had mentioned were forming in mitosis as part of chapter 11, these body cells are what we call diploid, and diploid refers to the number of chromosome sets. So when I say something is diploid, we have the prefix there, di. Di means two, okay? Like a disaccharide is a type of carbohydrate made up of two monosaccharides. Diploid body cells have two sets of chromosomes. One set came from mom and one set came from dad, and those are these homologous pairs that we just saw here on this last slide, right? These are my I'm looking at pairs of these. I have one set from mom and one set from dad. So those pairs we represent, we can say an individual or a cell that's diploid is represented as 2N. N in this case represents the number of sets of chromosomes. So in other words, let's say I have 2N, and in this case, I've got 23 chromosomes. I know that makes up one individual set. So in total, for a 2N cell, I have 2 times 23 or 46 total chromosomes. Human diploid cells, like we just saw here, have 44 autosomes. That's pairs 1 through 22, and then a final pair of sex chromosomes. So that means we have 2 sex chromosomes and 44 autosomes, or 22 pairs of autosomes. Diploid cells are what we create through sexual reproduction when sperm and egg join and fuse to create a fertilized egg and then a gamete and then a fertilized egg and then an embryo and so on. And that's kind of, what's used to create everything in that multicellular organism that's then created from that. Half of the chromosomes in a diploid cell, again, come from mom, and half come from dad. Now, the cells that we actually use, and we can see here, we have one pair of chromosomes, the purple and the green, and then another pair of chromosomes, the red and the blue. So maybe mom donated these two, up here, purple and red, and dad maybe donated green and blue. Okay, that's kind of how we could think about it. Gametes, on the other hand, like I just mentioned, are going to be different. They are considered not diploid, but they are haploid because they have half of what's present in a normal body cell. So for half, that means in humans, again, my N is equivalent to 23 because that's one set. And so if I have a haploid cell, we call that 1N, or like you see right here, just written as N. That single cell, that single N cell, would have to have then 22 autosomes and 1 sex chromosomes. We have no homologous pairs in our haploid gametes. Okay, so that means in sperm and egg, we do not see homologous chromosomes. We do not see pairing of any sort. The only thing that we're going to see is a total of 23 chromosomes. So that means in the production of these, we have to find a way to separate those pairs that we saw in this image back here, right? We're not going to inherit all this or pass all this down to our offspring. We only want to pass down half of what we see there. So if we look at the cell down here, right, I have half of what I had on this cell right here, which was diploid. So my gametes that are haploid, I end up with having half of that genetic information, or again, no pairings. I don't have 2 large chromosomes here anymore, or 2 small. I have 1 large and 1 small. Okay, and those are not homologous because they're not the same size. When we create a living thing, like we said on the last slide, that involves creating a diploid cell. And the way that we get a diploid cell is we take a 1N haploid cell and a 1N haploid cell, and we fuse them together. So we essentially create 1N plus 1N equals 2N, right? That is my diploid cell. That's what then is used to create every other body cell and other tissue type in a growing and developing organism, okay? So again, meiosis is what's going to be used to actually create these gametes that we're talking about here, the ones that are actually haploid, that contain for humans only 23 chromosomes in what we often are just going to call sperm and egg, okay? So meiosis, unlike mitosis, is going to have 2 rounds of division. In mitosis, we saw prophase, metaphase, anaphase, and telophase. In meiosis, we're going to see the same 4 stages, but we're going to do them twice. All right, so it's essentially the same general process. The events are going to be very similar. It's very orderly and organized. The difference is going to be that in this case, we have to do it twice because we want to make sure that we divide the cell enough times to have the number of chromosomes, like to cut them in half. So as part of this process, one of the things we're going to see, a couple of important things to kind of keep in mind here, and these are 2 bullets that are exceptionally important on this slide. Homologous pairs will separate in meiosis 1, and then sister chromatids are going to separate in meiosis 2, and those sisters are the only things that we saw separating in mitosis. So when in chapter 11 we talked about sister chromatids, remember those are the left and right halves of my X. They get pulled apart in anaphase, right? And then they get fully separated in terms of becoming part of different cells through telophase and cytokinesis. So the division of those sisters, the pulling apart of those in meiosis 2, most closely resembles what we saw already in chapter 11. So you can think of meiosis 2 as essentially being same as mitosis with a few slight differences. Okay, I like the diagram here at the bottom because I think it helps to distinguish between what's a sister chromatid and what's a homologous pair or a homologous chromosome. So homologous chromosomes, a homologous pair, what we would get from mom and dad would be, again, like one purple and one green. Same shape, same size. These obviously have gone through synthesis, so we've created sisters. That's the other half of each of these chromosomes. And those sisters you can see are being labeled down there. So within 1X, remember, each half is a sister. And then when I look at a pairing of similar chromosomes, those are what we use to call, or those are what we use the term homologous chromosomes to represent. All right, so as part of meiosis, we're going to go through what's happening in these stages here. So we've got meiosis 1, and then we're going to have meiosis 2. Again, same series of events that we saw in mitosis. We're going to, again, have prophase, metaphase, anaphase, and telophase, and you can see that here. If you want to jot down, what's actually happening in each of these stages, that may be helpful. I'm going to talk you through them, and you can see the descriptions are down here at the bottom. Okay, but I don't have a separate slide that, just outlines each of these separately in text. So meiosis 1 is going to occur during M. So if you think about what we learned about our cell cycle, right, we had G1, we went through S, G2, all of that was part of interphase, and then we had this other smaller piece of the pie that we called M. And in chapter 11, I mentioned that M is what we were calling mitosis, but M can also fit with meiosis. So if we're looking at germ cells, the cells that we store and keep in our ovaries and our testes, those reproductive organs, when we need to make gametes, okay, M is going to represent meiosis. M does not represent mitosis in this case, but it's the same cycle diagram. It's just now we're looking at M as being meiosis. So M is going to happen after interphase. So we still go through G1, G2, and S. We still copy our chromosomes. So we still start in our germ cells with 92 sister chromatids at the start of meiosis 1. First stage of meiosis, as we saw in mitosis, is prophase. Because it's part of meiosis 1, we call it prophase 1. Okay, Roman numeral 1. So in prophase 1, same series of events as what we saw in mitosis. We break down our nuclear membrane. That's the first step. We condense our chromosomes. So the chromosomes are going to pack together into those nice X shapes that we're able to see under a microscope. And then we're going to start to form our spindle fibers from the centrioles present in things like animal cells. So our little spindle fibers are coming into form here. They'll eventually extend, and these centrioles will move to opposite sides of the cell. And as we learned before, those spindle fibers are going to help to separate our chromosomes. So that's all prophase 1, same series of events as prophase in mitosis. Now, where we start to see some differences is here in metaphase. So metaphase, if you remember, means middle. And in metaphase, our chromosomes are still going to line up in the middle. Nothing has changed there. But if you'll notice how this appears to be different, in metaphase and mitosis, I had the purple, green, red, and blue all lined up right in order like this across the middle. Here, what we're seeing is I've got purple here and green kind of on top, right? And I've got blue and I've got red kind of on top. And how that becomes importantly different is that now we're not going to separate our sisters yet. What we're going to actually separate is the homologous pair. So my pairs are going to line up together. So purple and green line up together, red and blue line up together. And then when they get separated, my whole X is going to go to each of those poles. So I'm going to end up pulling in anaphase here my green and my red, the whole thing, not the sisters, but the whole homologous chromosome to one end. All right, and same at the bottom. I'm going to pull the purple and the blue, the whole X to the other end. What I end up with in telophase 1 then is 2, and after cytokinesis, the first round, which is not being shown here, but we can assume that's happened since now we have 2 separate cells. Telophase 1, I'm going to start to do everything backwards that I did in prophase. I'm going to reform the nuclear membranes. I'm going to unwind the DNA, and I'm going to break down the spindles. And if you'll notice what I'm left with here, I already have just half of what I started with, right? I had 4 X's. I had 2 homologous pairs in this one single cell, and now I just have 1, right, from those homologous pairings. So I don't have 2 big X's anymore in any cell. I have one of the big chromosomes here, the other one's in the other cell. And I have 1 small chromosome here, and again, the other one's in the other cells. So I've separated those pairs that we originally had in that first parent cell. So if you'll remember, we do need to continue to divide, though, because we don't want to end up with chromosomes with those extra sister pieces. Those are not needed for cell function. And so when we do this production of gametes, we also want to go through the round of sister chromatid division. We don't need to have those sisters together. We need to separate them also. And so what we end up making in this process is actually 4 haploid or gamete cells. So right after telophase 1 and cytokinesis 1, we go into prophase 2. And prophase 2 does the same stuff as prophase 1. So we wind up our chromosomes again, we make our spindle fibers, and we start to break down that nuclear membrane. Okay. Metaphase 2, notice how this appears different from what we saw on this last slide. Right here. So here they're lined up top and bottom, right? Both pairings are top and bottom. Now, if we go on to this one, we can see I don't have those pairs anymore. So this one, like I said earlier, reminds us a lot more of what we saw in mitosis, because now they're lined up in a way where I can actually physically separate those sisters. And that's what's going to end up happening. So metaphase 2, we still end up in the middle. Anaphase 2, we're still going to separate and pull these things apart, but now we're not separating our homologous pairs, we're separating our sister chromatids. So they get pulled in opposite directions. And what we end up with here is 4 entirely separate cells after telophase 2 and cytokinesis 2. They are uniquely independent, even though here they don't appear to be unique and different. If we think about in humans, we would have 46 of these little guys inside this nucleus. You know, the random assortment of all that can lead to some unique variety, okay? And so as part of that, we're going to end up with 4 what we call unique and individual haploid cells. So here's that whole process. I think this is the diagram that the textbook likes to use. You start with interphase, go through prophase 1, metaphase 1, anaphase 1, telophase 1, and then we scoot all the way back up here, meiosis 2, metaphase 2, prophase 2, metaphase, anaphase 2, and telophase 2. And so our end results are going to be different, right? We're not creating identical copies. We're going to create unique copies of cells that can be used for a totally different purpose, right? We're not trying to replace anything in our cells. We are simply we're simply trying to create some variation in offspring. All right, so here are kind of side-by-side comparison of mitosis and meiosis, and I really like this diagram. So I think it sums things up really well. A couple of major differences. Meiosis, obviously, we're going to divide twice. So we start with one parent cell. Notice the parent cell in both of these looks exactly the same, right? This is what we inherited from our parents. That first cell in meiosis splits once, and it's going to divide the homologous pairs. It splits again, and we separate our sisters. In mitosis, on the other hand, we simply separate the sisters. We never divide those homologous chromosomes, those pairs. Meiosis also creates haploid cells, right? These are my haploid cells. They have half of the DNA that we originally saw here at the start. So instead of having 4 chromosomes, they have just 2 chromosomes, right? Per cell, I should say. We also have a scenario here where in mitosis, we don't pair up these things called our homologous chromosomes. And I failed to mention that back here. So let me scoot back real quick. One of the things that I needed to add here that actually happens in prophase 1 is that these homologous pairs, like what you're seeing here, the purple and the green, and then the red and the blue, they actually do one additional really important thing here in prophase 1, which is that they do this process called crossing over. And crossing over is pretty unique, and it's only going to happen in germ cells during this process. But what it's going to do is it allows for the exchange of genetic information that normally wouldn't be exchanged because it's coming from 2 different places. So what I'm talking about here is, like, let's say we're looking at the purple and the green chromosome. When we cross over, what that essentially means is the X's are going to kind of lay on top of each other in a way. At least part of them is going to cross. And when that happens, when they physically touch like that, they can exchange genetic information. So what was once on mom's purple chromosome can be added to dad's green chromosome, and vice versa. What was on dad's green chromosome gets swapped with what was on mom's purple chromosome. And so you have the potential here of creating new genetic combinations that have never been seen before in the history of everything human by simply mixing those 2 genetic lines, okay? So again, that crossing over process happens specifically in prophase 1. And we can see that, by the way, happening right here. So see how their legs are kind of flipped over on top of one another. And as a result of that, we've exchanged. So we have a little bit of yellow here on this red chromosome, and we have a little bit of red here on this yellow chromosome. All right. So that would be different. We never cross over as part of, we never have crossing over as part of mitosis, only as meiosis. All right. So leading into this then and looking at crossing over one more time, we know that when this happens, there are maybe increased likelihoods of exchanging genetic information when that genetic information is maybe farther apart. Okay, so this leads us into this conversation about what we call gene linkage. Genes that we have that are tending to be very close together on a chromosome tend to always kind of stick together. And the reason for that is that it becomes very unlikely that we actually cross over right in between them. All right, and what that means is that if we've got genes and we have thousands of these genes in our bodies, but per chromosome, we could have hundreds of genes just on one chromosome. The closer together those two genes are, maybe they are direct neighbors, right? They're sitting right next to each other on that chromosome. In order for them to be separated through crossing over, the other chromosome would have to come in and like separate them right here, right? It'd have to cross over right in between those two genes. And we know that that's not very likely to happen. Sure, it has to cross over somewhere, but in general, the closer two genes are, the more likely it is that they remain together, okay? And so when we talk about this, we use the term alleles, which represent different types of varieties of a specific gene. So we could say blue eye color and brown eye color. Those are alleles of the eye color gene. So crossing over that exchange of genetic information is certainly likely, and it generally happens with every single division in meiosis, with every kind of round of production of these gametes, these sperm and egg. But it's more likely to happen when you have alleles for traits that are further apart. That's where you get that mixing. If they're super close together, it's not likely that they get separated, and it's not likely that that genetic information gets exchanged. Okay. Again, as I mentioned just a few minutes ago, this occurs again only prophase 1 of meiosis 1. And again, we can create lots of really cool and unique combinations of genes through this process. Okay. All right, so that's what I have for section 12.4. As always, ask me if you've got any questions. Otherwise, I will see you next time. Bye, y'all.